Okay, sorry, I was muted. Okay, then I'll talk about a subject a little bit less fun than astrophysics, that about cancer actually. Then I'm Tarcisio, I'm from the Swiss Institute of Bioinformatics along with Frederic Bastian. One of the use cases that we have done the project about cancer biomarker research, to be more precisely. Then I'll give you a little bit of introduction about first of all, what's cancer? Cancer is actually an uncontrolled cell proliferation that results in a malignant tissue growth. Essentially, our immune system are not able anymore to eliminate this abnormal cell that results in some cancer type, such as a breast cancer. And to characterize these cancer types and also to identify some therapy strategy, specifically to a cancer type like a breast cancer, for example, we can use biomarkers actually. Biomarkers are indeed substances used as an indicator of some biological state. And this biological state could be a cancer type, such as a breast cancer. And we can have like a molecular biomarker, for example, such as a gene biomarker or a clinical exam image, for example. In this slide, we have for the breast cancer here. Then another major component when you're doing cancer biomarker research is the gene expression. Gene expressions is actually the process by which the instruction in our DNA are converted into some functional product, such as protein. This process actually can be divided in two main steps, like transcription and translation, where the genes, part of the sequence DNA actually are transcribed into a messenger RNA. This messenger RNA goes out the nucleus of the cell and are then translated into proteins that actually carries the function of a gene. When we measure the gene expression levels actually in different types of conditions, such as cancer tissue and healthy tissue, then we can see that these gene expression levels can vary, actually can increase, decrease. And based on it, we can also derive some gene biomarkers, for example, based on the gene expression as well. In the case of the iNode project, actually, we had a question from a cancer biomarker research that essentially he would like to know actually which were the top genes differentially expressed in breast cancer that are also cancer biomarkers by the Early Detection Research Network. Essentially, when this kind of question are the question that the iNode systems currently can address, based also in the knowledge that we have about cancer biomarkers from the OncoMix database, a database for cancer biomarkers actually. Then essentially the iNode system, it's able, it retrieves actually one gene that was the MAGEA6 gene. And when we look at the early detection portal, research portal, actually, we see that the MAGEA6 gene is actually the quality assessment state for breast cancer is under review. Then it's also indicated as being a breast cancer biomarker. And based on the differential expression data that we have from the OncoMix database and through the iNode system, actually, we can see that this suggests that this biomarker actually is a good candidate for breast cancer, for indicating breast cancer actually. Further analysis actually, when we look another database that also part of the iNode projects is the BG database that contains gene expression for healthy tissues. We can see that this same gene actually is reported to be absent in this anatomical entity called the thoracic mammary gland, actually that's located in the breast. What further implies that this is a good candidate to be a biomarker. Then we can see that currently the iNode system also can already give really good insights for the researchers actually. Other type of question that the iNode system it's able to answer that I mentioned here, also because in the further presentation, you're gonna see it is part of the demos. Then thank you very much for your attention, and I will hand over to the next presenter.